Thank you.
Recording in progress.
All right, so I'm just gonna pull up the kidney models. I think we'll start with that. So we'll kind of go through a few of these models and then we'll get going into kidney lecture side of things. And I'd like to do some practice questions as well today if we have time for that. Okay. All right, so remember for your final, right? You guys have muscles. We've already talked about the arm, the leg, the torso models, SI, right? So we've done all of your models here in SI. also have your kidney model as well, in addition to all the previous models that we already talked about: sarcomere, brain, spinal cord, eye, ear, right? So make sure you look at all of those. And where was I going? Oh, and don't forget to do your packet, right? Your muscle packet, origin, insertion, action, innervation. Coloring in the muscles might be a good idea on your packet, because on your lab exam, they will be tested colored in, right? All of the letters will be whited out, so you can't see the letter, but they'll be colored in. So I recommend coloring it in. You don't have to color it in to get points, but I recommend coloring it in. Okay. Are there any questions at all on your packet, or anything about your exams coming up?
Can you hear me?
Yes.
What do you know what muscles of the neck are going to be tested on the on the torso models, like the big models?
Yes. Should just be sternocleidomastoid for the big torso models.
Okay, thank you.
The other muscles of the neck would be in the packet that we would test you on those.
Okay, thank you.
Yeah, no problem.
And I just need Maria to type into the chat her first and last name and Delta ID.
And so remember, on this neck here, like I was saying, this would be the one we would test you on for the packet. Sternocleidomastoid is the only one you would know on the actual torso models. And you can see that here. It's very obvious muscle. It's already pre-labeled on this packet. So we really wouldn't ask you on the packet. We would be looking for that on the torso. And then obviously you don't have to do origin, insertion, action, innervation on any of this stuff over here, because it's not muscles, right? Like all these arteries, the larynx, all of that. So you're just dealing with the muscles on the neck. Okay. So no questions on that. Everybody's good. All right. So let's look at the kidney model, and then we'll go into the kidney lecture. And then depending on how much we do today, I don't know if I'm going to finish it 100%. I think I'm going to leave the last little part for Thursday. And then maybe if we can get a little bit ahead of the content, like maybe do male reproductive and get a little ahead, then that way next week I can help you guys review for the exam, if you guys are good with that. So if you have time this week, try to get ahead. I know that urinary lecture is the one assigned for this week. Try to watch that and a male reproductive, so that we can kind of move a little bit forward. Again, so next week I can help you guys with reviewing. Okay. All right. So once again, you guys have access to a video of the models.
Question?
Yeah.
So in terms of like the muscles and the muscle packet, now he's going to say, for example, show us a muscle that's on paper, right? And then he's going to show us the muscle in real life, but those are not going to be similar, right? Like he's not going to ask us like, what's this on the arm, for example, and it's not going to be where, like, there's certain things on the muscle packet that are not on the muscle model, and there's certain things, right?
Yeah. The best way to kind of think about it is, what is my list that shows me my expectations for the muscles model wise? And then what do I know all of those on the actual muscle models? And then what is my list of what I need to know on the packet? And do I know that on the packet? So overall, you should be having 99%, but I do know what you're talking about. There's a few kind of random ones that will be specifically on the packet and not tested on the muscle models. And you would be right. Those will just be, we will have the exact page from your packet on the test colored in, and we'll ask you that on the packet.
Okay. And I don't know if this is true. I don't know if I'm making this up, but I remember when we first got the muscle packet, he said that we could look at his colored ones to check if we had the correct coloring. Yeah.
I think what he meant was the correct answer, which I already gave you guys in that Guide to Muscles link. So in the past, it was like students would do all the work and kind of fill it in and then go up and check their answers. Personally, for me, that's why I shared with you guys who come to SI the answers. I didn't feel like that was helpful for me. It was like, I felt like, okay, well, now I just wasted my time thinking it was this muscle and now it's actually not. So that's what he should have been talking about. There's no like right way to color it in. If when you're coloring it in, you can just kind of trace the muscle and it should work. I colored them in when I was in the class. I felt like it really helped me. And the number one tip I would just give you guys for studying is pull out the arm muscle model and your page in the packet and go over them side by side. I know that there's a few that aren't on one or the other, you know what I mean? But overall, it should be almost exactly the same, and it will help you learn both. You know, you'll be looking at it on the packet, and then you'll realize, okay, that's the same muscle over here, and vice versa. That's what I did on the muscle final, and I felt like that was really helpful for me. So that's what I would recommend.
Okay, thank you.
Yeah. Any other questions? Your final lab exam should be 140 questions, somewhere around 100 on the muscles and like 40 on the models. But this time everything's kind of mixed together. So I don't know an exact answer about like 140, but I do know it's 140 questions. So that's that. And then your final lecture exam should be either between 160 and 170. I think it's 170 questions. So if you kind of had a few moments in this semester where you're not super happy with your grade, the 170 question final can be enough to boost your grade. If you feel really good because you have a good grade right now, remember that 170 could bring your grade down, right? So I just encourage you to keep studying. Don't give up. You're almost there. It will be worth it. So yeah, the one thing I would say with the final, before I get into this, I just want to give you like a general advice. The final exam is the best way to study for the final, in my opinion, is to make sure you understand like the organization, like kind of how I do the when we go over it in outline form. Understand the organization, because a lot of the questions are like basic, and I'm putting that in quotes because it's a lot of like, you know, which of these is a part of the structural nervous system? Which of these is a part of the functional nervous system? You know, and you might get in the weeds talking about like, how does the sympathetic nervous system work, which may be a question, but a lot of the questions are more like, do you even understand the organization of the whole chapter? So the more you guys make sure you go over and over like the memorization stuff, that will help you. So hopefully that kind of helps direct you. Kind of transitioning into kidney. So when we talk about kidney is going to be the hardest topic of your exam. So when it comes to the actual physiology part of this lecture, it's kind of the most complicated. But it's not that bad. This and muscle contraction are going to be the areas that if you don't study them, you will not be able to answer questions on them. So the process of muscle contraction, the process of a synapse, how does one neuron communicate with another neuron? And then what we're going to talk about, what I'm trying to save for Thursday morning, the 3 physiological processes of the kidney. Okay, so those are kind of your main really hard points. If you don't kind of get it all, you're probably not going to be successful on the exam. Okay, so looking at the kidney model kind of helps us with the kidney lecture. So the kidney is set up into kind of 2 structures here. We have the outer cortex, which is like this part here, this outside part, it's like a light pink color. This light pink is called the cortex of the kidney. So cortex. Oh, that got really far away. Okay, you're all with me. And then the inside part is the medulla. So again, cortex would be the outside, and then the inside would be the medulla. So that'd be like this kind of hot pink color. So this whole little layer here is the medulla. And the medulla is technically where the pyramids are, and each of these little hot pink triangle things, each one of those is a pyramid. And if you remember, a pyramid is a triangle, right? So that makes sense. Pyramid. And within all of the pyramids, we have nephrons. So this guy right here, kind of see it? That is a nephron. And we're going to relabel that again on another model. So nephron, that's the functional working kidney, working unit of the kidney. It's what filters blood to make urine. Then in this little center part, you have the renal pelvis. That's like where everything kind of comes together. And then this would be the ureter, this white tube that leaves. And then that would drain down into the bladder. So ureter would connect down into the bladder. So I'll put B for bladder. We have the renal artery and the renal vein. Renal artery. I'll just put vein because I think we all know. Blue would be vein. Red would be artery. Renal artery, renal vein. And then you have your minor and major calyces. On this model, you can see the minor. So I'm going to try to circle it. They're really visible here. You see this like kind of little V shape? One there, one there. Can you see my, okay, yeah. Okay, this little V shape. That is a minor calyx. Like we've talked about multiple times, there's a plural form in Latin. So multiple calyxes would be calyces. And then they flow and join together into the major calyces, which are kind of the same shape, right? Again, kind of like a little funnel. So those are major calyx. Okay, any questions there? All right. Okay, so moving on to this model. So what you're going to see with the models is you have this one right here, then this one right here, and what you notice is this guy is the same 3 as this one. So they're almost the same thing. Things do look a tiny bit different, so I recommend going over both, but just don't get super stressed out. We will use either or both on the lab exam. So I would go over both of them. We could also use this. Again, it's going to be the same stuff again, and I'll go over them. And then the last model we use is this one. It has 4 different sides on the bottom here. And I'll show you guys that one too. So let's start with this guy. So again, what we're going to see, and then I'm going to transition so I can actually see it on the picture. Remember how I just said, this is the cortex of the kidney. You're kind of like trying to orient yourself to what you see. Here's the cortex and the medulla. And I told you each of these is a pyramid, remember? So this is a pyramid zoomed in. Then there's these little glomerulus section, and that is a glomerulus zoomed in. Does that make sense? So these models is zoomed in versions of each other across the model. So first, we're going to talk about the kidney again, and then we're going to go to the pyramid, and then the glomerulus. Okay. Okay. So again, our outside portion here, I'm going to just put, well, I'll just write it. Cortex, right? Cortex would be this outer region that's kind of this light orangish color here, cortex. And then this row, right, that whole section, anywhere those pyramids are located would be the medulla. So this is one pyramid, and the pyramids, or that dark, that the medulla is that darker burgundy kind of color. Make up medulla. So then we have the minor calyces and the major calyces. Minor would be right here, kind of these little white funnel looking shapes. So those are minor calyces. And then they drain or flow into major calyces. So this would be a major calyx is one, calyces is multiple. And those all flow together into the renal pelvis. Then we have the renal artery and the renal vein again. And then remember that the renal pelvis flows out of the ureter, and the ureter connects to the bladder. This over here, if you were, you could be asked on the test, that would be a nephron. And then you have your collecting duct, but likely not asked on this part of the model. We're going to ask you on the next one. Got it? Everybody good? Okay. How can you tell the difference between the medulla and calyx major? Oh, that's a good question. So if it's asking for medulla on the exam, likely we will put some type of, how we use the tape? We would use a piece of tape and stick it across this whole thing. And then the wording of the question would be, what is the name for this region of the kidney? Is that clear? What is the name for this region of the kidney? Versus what is the name for this structure? Structure would be pyramid, right? The region would be medulla. So just pay attention to the wording on the question. In terms of major calyx, the arrow would be directly pointing just down, right? So if my piece of tape, my arrow, was pointing just at this, then you know that it's the major calyx. Does that answer your question? Do you feel like that would make sense on the test? Okay.
What?
All right. So then we go one step closer. This now is just one pyramid. Does that make sense? So you kind of look at it again. The pyramid is the part down below. Well, it's kind of like just one pyramid. It's like one little cross section. So this is still the cortex. This down here would be medulla / pyramid, right? The part down below. Now, there's nephrons here, right? We have our nephron, which is the functional working unit of the kidney. There are two different types of nephron. There is what's called the juxtamedullary nephron, which is this one. Juxtamedullary nephron is basically when the loop of Henle is deep into the medulla. Another way of thinking about the juxtamedullary, it's probably simpler, is when most of the nephron is in the medulla. So if you look, this is juxta, which means near, right? Juxtamedullary, near the medulla. So most of that nephron is in the medulla. Versus on this side, this nephron is called a corticonephron. And a corticonephron is when most of the nephron, this is important to know for the test, is in the cortex. So this is something you want to know for the lab and the lecture. Technically, the lecture definition is the lecture definition is very similar. It's almost the same. All right, so cortico and juxtamedullary, so you would test on those on the actual exam. And then those flow into the loop, the collecting duct. So this is the collecting duct. The collecting duct then hooks up to the calyces, which are those funnels that I showed you before. And then you have the arcuate artery and arcuate vein. Okay, arcuate artery, arcuate vein. Okay, any questions here? One more thing I can show you. So you have your actual little glomerulus, which is where the filtering of the blood to make urine is happening. As you guys can see, here's my blood coming in, right, from my renal artery, and it flows into my little glomerulus in order to filter the blood to make urine. Urine will then, this is actually really cool that I chose this yellow color. Urine will go here, down the loop of Henle, up the loop of Henle, over, I'm telling you this so that I can show you what all this is called, over, and then down into the collecting duct. And then where does the collecting duct go? The collecting duct will go to the calyces. First, it will go to minor calyces, then it will go to major calyces, and then it would be worth it for you to write this down because this will be good for you to know for lecture. And then after major calyces, it'll go to the renal pelvis. And then what did we say? Then it goes to the ureter, to the bladder, and then out the urethra. So as you can see, this would really be the path of urine. So the true path of urine would be blood gets filtered in the glomerulus. Blood passes first through the proximal convoluted tubule. And I want to label this for you guys. Then it goes into the descending limb of the loop of Henle. Then it goes to the ascending limb of the loop of Henle. Then the distal convoluted tubule. Then the collecting duct. Then the minor calyces, major calyces, renal pelvis, ureter, bladder, and urethra. So there's your whole journey of urine, okay? All right, so labeling over here, this would be your PCT, proximal convoluted tubule, and you do need to be able to label this for the lab exam. This would be your descending limb of the loop of Henle, descending limb of loop of Henle. Again, you need to know this for the lab exam. Ascending limb over here. Let me try to get rid of, let's do a different color, make it stand out. Let's do pink. So again, ascending limb is this side here. As it ascends, that's the ascending limb of the loop of Henle. Then it gets into the distal convoluted tubule right here. So I'll do green. Distal convoluted tubule, DCT, and then out to the collecting duct. So you need to know that PCT, DCT, ascending and descending for the lab exam. Good? All right, any questions there? All right, so now we have our glomerulus, just the glomerulus, right? Looking back at the overview, we looked at the outside of the kidney. We looked at just one pyramid and cortex, like over here, right? Then now we're looking at just one glomerulus, which is like this little ball. Does that make sense? So it's very zoomed in. When I look at that, this kind of gives you the story of how the blood is filtered. So it comes in through the afferent arteriole, leaves the efferent. Remember, aff means towards. So over here, this is the afferent arteriole towards the glomerulus. Then we get the efferent arteriole, leaves the glomerulus. Then we get the proximal convoluted tubule. Remember that once the blood is filtered, it goes to the proximal convoluted tubule first. You can also think of it as proximal is proximal to the glomerulus. The like bundle of capillaries here is called the glomerulus. It is made of fenestrated capillaries. And then it sits inside of the Bowman's capsule, which is like the outside of it, like the bowl that it sits in. You can think Bowman's bowl, sounds like a bowl, and that's the glomerulus bowl. Bowman's capsule, renal capsule, renal corpuscle. Okay, so it has some different names. You want to know the different names. And then over here, this is our distal convoluted tubule. In the previous picture, you would have seen that kind of those distal and proximal sit next to each other on the actual anatomy of it. So distal convoluted tubule, again, is not technically like immediately connected. It's distal to it. But on this model, we put it together. And there's a reason we do that. So it's really important that you guys know this for the lab exam as well. So on the bottom of the distal convoluted tubule, you have specialized cells that are part of one of those physiological processes. If you guys haven't watched the lecture yet for kidney, I would highly recommend you do that. So the macula, wait, something looks weird. Is that right? Macula densa? Something sounds wrong there. Macula densa. I think that's right. Someone correct me. Am I saying that wrong? Macula densa. Okay, good. Sorry, I took my final. I took a final yesterday, and then I took another final today. So I think my brain might be a tiny bit fried. So macula densa, these are the specialized cells that line the bottom of the distal convoluted tubule and release renin. Okay, so this is part of the renin angiotensin aldosterone system, which is very important that you guys learn. And together, we're going to talk about that, the renin angiotensin aldosterone system on Thursday morning. Together, the afferent arteriole, the macula densa, and the efferent arteriole, we're going to number them. Number 1, number 2, and number 3. So number 1 is the afferent arteriole. Number 2 is the macula densa. Number 3 is the efferent arteriole. Together, 1, 2, 3 make up the juxtaglomerular apparatus. So that is super important that you know. So it's the juxtaglomerular apparatus. That is, the function of that is to basically like regulates blood pressure. All right. Okay, so that's that for that view. Any questions there? Okay. All right, so let's go ahead and erase that. And then I'm going to show you the other model. I don't think I'm going to go over the same parts again. I think I'm just going to show you what's different. So this model would be pretty much the same. You guys can see that, right? See how this has a
Grayson.
cortex, pyramids, minor calyces, right? Major calyces, the renal pelvis, the ureter, renal artery, and renal vein. So you can pretty much see everything is the same. The big thing that's different on this model is that this is the adrenal gland here. So on the other model, we didn't have an adrenal gland. This model has it. The adrenal gland is the same as the kidney. It has a cortex and a medulla. The medulla is on the inside. The cortex is on the outside. You do need to know that for the lab exam. Okay. What is the overall entire thing? Adrenal gland. What is it made of? The cortex on the outside and the medulla on the inside, just like the kidney. All right, moving on to this one right here. This again would be the same stuff we were already looking at on this model as on the previous model. This model you can see again the juxtamedullary and corticonephrons. So I'm going to label both of those. So this would be a juxtamedullary and this would be a corticonephron. So corticonephron, juxtamedullary nephron. And in case you're wondering, because technically my pointer is like pointing to a descending limb of the loop of Henle, you know, questions that come up is like, how would I know if it's pointing to that on the exam? You know, it's going to ask you what type of nephron is this? So then there's no doubt in your mind, you know what it is. If we're actually looking at what my pointer is pointing to on the right, that would be the proximal convoluted tubule, right? It's still the part that's curvy. And versus over here on the left, if we're looking at my pointer, that would be the descending limb of the loop of Henle. Does that make sense? What's the type of nephron it's a part of? I labeled that versus the part. They flow together into the collecting duct, right? We have the PCT, DCT, ascending and descending limb. Collecting duct. I used to get confused when I was in the class, how do we know which one is descending limb and how do we know which one is ascending limb? Descending limb goes away from that glomerulus. So on the exam, find your glomerulus, then you know that proximal convoluted tubule is the one next to the glomerulus, proximal to it. And then the descending limb goes away from the proximal because it's getting to its destination, which is out of the body, goes to the ascending limb, out the distal convoluted tubule to the collecting duct, to the minor calyces, right? This would be a major calyx down to the renal pelvis, the ureter, the bladder, and then out the body through the urethra. So this is a major calyx. Any questions there? So I went through it faster because I feel like it's just kind of a repeat of what we just said. Arcuate artery, arcuate vein, I already labeled. They're these blood vessels here that go across, right? Arcuate artery, arcuate vein. Good. Okay. All right. So looking at the next part of this same model, this is going to be the same thing again. Afferent arterial, glomerulus, renal tubule, Bowman's capsule, right? All of the exact same stuff. I don't think it's necessary for me to relabel that guy. All right, so that was 1, 2, 3 of your kidney models. So this is your last model, the kidney model, this one here. There's also this model, and I'm just going to show it to you before we look at the other one. This model doesn't have a whole lot on it that you would be tested on, but I do want to just kind of show you what could be tested on it because we could use it. And I believe I had it on my final. So renal artery and renal vein, okay? They would come from your descending aorta and inferior vena cava, which then branch at the bottom there into the femoral artery and vein. So this is the, inferior vena cava and descending aorta. And those branch into our iliac artery, iliac vein here. And what you can see on this model is the outer cortex that we already talked about, our inner medulla made of the pyramids, right? I'm going to put medulla / pyramids. Then the renal pelvis, where everything kind of comes together. All of that urine getting made comes together. And then it flows out of the ureter, right? So this is your ureter. Kind of like a recap of what we've already gone over. This is your adrenal gland. And then this is the bladder. There's a few structures on the penis, but those are going to be more in line with our reproductive lecture. So I'm going to save that. But there's a few things like prostate gland, seminal vesicle, and the different types of urethra. But we're going to talk about those with male. But everything else on here, for as far as kidney goes, you have that for this model. Okay. All right. And like I said, I'm hoping to do some review next week. So this model gets closer and closer. So first we look at a nephron. I'm going to go back. First we look at a nephron, then we look at the glomerulus. This is not in order because this would be outside. And then this would be our medulla. So I'll try to go in order. So this would be the furthest away, the view furthest away. Same stuff we've been saying. I'll just write C for cortex on the outside. M / P, right? It's the medulla / pyramids. Each of those would be a pyramid. You can see your calyx, major calyx there. This is the RP, renal pelvis, and the ureter. You're not honestly likely to get tested on this model on this view. So, you don't really need to stress about it, but I already showed you, what we could put on there on this model. Just unlikely that it would be on there because there's another angle of this model we like to use for the test. So I'll show you that right now. Okay, so the part that we like to use pretty consistently every semester from this model on the test is we really like this side. And that's because it shows you the two types of nephrons really well, and it shows you the blood vessels that supply them. So again, this would be arcuate artery, arcuate vein. I'm going to label that. Then we have our two different types of nephron. Remember, there's two different types. This is going to be which type, and this is which type. So I'm going to put option, let's number them. Okay, so this is number 1. So is number 1 A, B? So you have your two options. So let's all answer, is 1 A or B? Is it a juxtamedullary or a corticonephron? Good. So it's cortico. We know it's cortico. Why? Because most of it is in the cortex. So obviously we don't need to label the other one. We know that the other one would then be juxtamedullary. So this is a corticonephron. This one over here is your juxtamedullary nephron. If this, well, we wouldn't label it, but just so you know, you see this nephron right here, that is not possible. The loop of Henle always has to be down in the medulla. I'm not really sure why the manufacturer did that. Maybe they thought, I don't know, maybe they thought that it would help the students to see something that was wrong, to talk about it. But this one is not real because the corticonephron still has the loop of Henle in the medulla. Okay, so cortico over here, juxtamedullary on the right. All right, last thing. So we have around the proximal convoluted tubule and the distal convoluted tubule, we have some blood vessels. So the blood vessels that are around the proximal convoluted tubule and the distal convoluted tubule are called peritubular capillaries. Okay. Now the blood vessels that are around the loop of Henle are called the vasa recta. Blood vessels are around the loop of Henle called vasa recta. So for this model, we really like this view because we can ask you 4 things really clearly, that which type of nephron, cortico and juxtamedullary, and then the 2 blood vessels, peritubular capillaries, vasa recta. the question would ask, what are the name of the blood vessels found in this location? What are the name of the blood vessels found in this location? Not describing to you around PCT, pointing you to that spot. Does that make sense to everybody? Are we clear on that? Okay, I'm going to erase. I'm going to move on here so we can check out the last view of this model and show you the last two views, I guess. So there's this one. This would show you proximal convoluted tubule, distal convoluted tubule, proximal being the yellow one. I'm just going to put PCT because I think we kind of all get it now. Distal would be the white. I just know because I see the yellow coming out of that one. So DCT. Which one is this? This would be the descending limb, right? Descending limb of the loop of Henle. Why? Because it's descending away from that glomerulus. It's trying to get that urine out, right? So ascending limb, ascending out. These three little circles are to show you that at the bottom of the loop of Henle, it's more permeable to solutes, because you guys are going to learn about that, right? Like water and sodium can pass through it easier. So that's why it shows you that it's like that at the bottom, like more spaced out. The cells are more spaced out. They allow more solutes to pass through. But we won't test you on that. So what we're looking for if we use this one is PCT, DCT, descending limb, and ascending limb. Okay. Questions there? You guys have questions, please chime in. Okay. All right. Last part of the model here. Okay, so this one is the same thing I labeled before, but the weird thing about this model is it kind of looks odd. So this would be your afferent coming in, and this is your efferent going out. So again, the reason it looks odd is because they're all the same color, but what you should know is that afferent is always on the left, efferent is always on the right. So this is efferent arterial over here, afferent over arterial on the left. If you're kind of wondering, how do I know, for the test, it should be if it's on this side, it's afferent, and if it's on that side, it's efferent. But we should be pointing only to the top structures, really, to have you distinguish it. If we're pointing here, really, we're more directing you towards the glomerulus. So we're looking for afferent, efferent. Your professor should be labeling that at the top. And if it says, what it's going to say is like, what is the name for this structure? And what we're looking for is glomerulus. So the glomerulus filters blood, right? So how does it filter blood? And I'm kind of segueing into lecture, which we're going to go into right now. But like, this is my capillary. So how does it filter? Here's my blood. How does it get, you know that when you make urine, you have water and like, let's say you have water and sugar in your urine. So how does that get out of the capillary? Well, we've talked about it before, right? We said that that's when the capillary goes through diapedesis and changes and has holes in it. Everyone remember that? And then that allowed things to get out, like white blood cells. So that is called what? Fenestrated, right? Fenestrated is when it has the holes. So if we ask you on the test, what is the name for the type of capillaries found here? Your answer would be fenestrated, okay? If I ask, what is the name for the holes in the capillaries? The holes themselves are called fenestrae. So every hole is a fenestrae. This hole right here, that gap, is a fenestrae. Fenestrated is the type of capillary, okay? And then that whole thing is, again, going to be sitting inside of the renal corpuscle or Bowman's capsule, which you could think of it again like a bowl. Bowman's capsule, like the bowl it sits in, also called the renal corpuscle, renal capsule. And then it flows into the proximal convoluted tubule next, right down here. Proximal convoluted tubule. No, no one can read that. Let's do black. And then up here at the top, this guy is the distal convoluted tubule. And we said that the specialized cells that line the bottom of the distal convoluted tubule produce renin and are called the macula densa, right? Macula densa make renin. Together, afferent arteriole, number 1, distal convoluted tubule, macula densa, number 2, efferent arteriole, number 3, are the juxtaglomerular apparatus. On the test in the past, when we've asked this question, we put a sticker with a number, and we put 1, a sticker with a number and put 2, and a sticker with a number and put 3. And then the question says, what is the name for 1, 2, and 3 together? Okay, so again, afferent, efferent, and macula densa together are called the juxtaglomerular apparatus. So on the test, that would be labeled as 1, 2, and 3. All right, do you guys have any questions at all on the kidney models? So again, make sure you look at multiple models because we do test you quite a bit on the kidneys on the lab exam, and you are going to see probably all of the models at least one station, which would mean a minimum of 4 or 5 questions on each one. So make sure you go over all of them. All right, any questions on that before we get over into lecture? Okay, so just a reminder, just depending on how much, because I'm sorry, I've been doing this lately and my mind is just, I think, moving too slowly for me. Depending on how much time we have today, I may or may not do practice questions, but a reminder for everybody here, Study Resources has really great practice questions for you guys. There looks like there's quite a few for this content as well. I recommend you guys look at it. If you haven't yet, check out YouTube videos and helpful links. These are videos that have helped previous people study in the class. There's a lot of videos for all of your different topics for the whole semester. And, if you haven't looked at it yet, maybe look at it now. It's helpful stuff. Like for muscles, I love this video right here. It was extremely helpful for me personally. Muscles.
Romeo and Juliet.
It kind of like shows you the actual right here. See if we can mute him. Can everybody see that? A little bit small. I want to show you. So he walks you through, if you haven't watched a Crash Course, it's really good, but he walks you through how the actual muscle contraction looks. Like I said, this is kind of a complicated thing. So I would recommend watching some videos to kind of help you visualize it, some other videos. This video I highly recommend for the cranial nerves. I think I forgot to show you guys. I really liked it. It is set to the tune of like a song, but it shows you like different people doing the stuff while the guy sings. I know when I was at this point in the class in anatomy, I was feeling kind of tired of studying. So I liked watching like a funny video and it helped me remember the cranial nerves. And this is a tangent from kidney, but on your final, I think I had like 8 or 9 questions just on the cranial nerves. And I was like very surprised, but pleasantly surprised because I had studied them. But you make sure you need to know your Roman numeral, the name of the cranial nerve, and then its basic function. If you learn the functions in this video, which is what I did when I took my test, you will be successful on the cranial nerves on the exam. So I studied cranial nerves with this video, and I was successful on the cranial nerve. So it's it's 3 minutes long. I watched it like while I drove. I watched it, I think, like 10 or 15 times. While I would drive, I would listen to it, and it kind of just helped me learn them. Okay. So that may be helpful for you. Okay. Everyone good. So last time we finished senses. So in this lecture series, we've done muscle, like contraction, nervous tissue, which is like the organization of the nervous system plus neurons. We talked about the brain. We talked about the spinal cord. We talked about the autonomic nervous system. And then last week, we talked about the senses. So there's a lot of content on this exam. And I would say people, kind of struggle with the whole nervous part, but don't neglect the kidneys, especially the physiologic part. I would just encourage you, don't leave any topic out. There's a lot. So if anything, study everything at less of a depth than maybe previous exams versus neglecting one topic. Unless it's like a process, like I was mentioning to you guys, muscle contraction. That would be one where you want to understand it in depth, because if you don't understand it in depth, how are you going to be successful on the test, right? Because it's kind of getting the whole thing. So you want to get that. You want to get the synapse, like really understand the synapse in depth, understand the physiological kidney functions, which we're going to talk about Thursday, which is like the loop of Henle, the juxtaglomerular apparatus, ADH, stuff like that. So those would be the areas that I would say are like the hardest to get, and you need to go over those for the test. Everything else for the final, you can go over it trying to understand everything in a simplified way, like we do with the outlines, versus like stressing super, super hard. So the mistake I made on my final, and I just am trying to help you guys, is I remember like spending hours on the brain lecture because it was so confusing to me. And then I got to the final and I was like, oh, like you just wanted me to know like the function of the diencephalon and where is the diencephalon located and what is its basic function. And instead, I was out here in my notes like, okay, so the diencephalon does this, and like here's the diencephalon. I'm like, okay, so it has all these nuclei, and then there's the nuclei, and then there's all these nuclei, and then I need to know like, okay, each of the functions. And it was like I was overcomplicating it, when really what you need to know is like, what is this? What is its function? Where is it located? How does it fit into the picture? If you do this in outline form, you should be able to be successful. But don't neglect understanding stuff that, like, if you don't understand it, you can't test on it. So I don't want to, like, mislead you, but, like, go into detail on these topics I kind of wrote down, and then try to get your basic idea of everything else without losing stuff. So hopefully that was helpful and not helpful. Like, for the senses, know which one is the actual dendrite. Right? Which one's the chemoreceptor? Which one's the mechanoreceptor? Which one is, et cetera, the type of receptor, and how does the receptor get triggered? That's what you should know for the senses. Versus like, I was out here like, I don't know, I'm always out here adding too much detail. You don't need to do all that detail. Just try to like know your basics. Like, what is happening? Where is it happening? What are the key structures? What is the key thing that's going on, instead of like all this too much detail.
It doesn't have any staples in it.
Sorry, I was trying to, hopefully that was helpful. I wasn't trying to get off on too much of a tangent there. So for urinary system, this is also called the excretory system because you are excreting. It's like your body's primary way of excreting stuff from the blood. We know that it filters the blood. I actually feel like before anatomy, I honestly didn't realize that the kidneys filtered the blood. So maybe that's new to you too. So they filter the blood, and then they balance pH in the body. So they help your blood not get too acidic or too alkalinic, like we talked about last unit. It can only be between 7.35 and 7.45. And if a patient has it and even 1 point out of that range, technically they would be acidotic or alkalinic. And then they balance blood pressure, which is the one we will talk about quite a bit through 3 different mechanisms. We know that there's the nephron, the functional unit of the kidney, the glomerulus. I'm gonna see how let's see if we can make this a nice outline for you guys. So we have our macroscopic view of the kidney. What that means is like when you look at a kidney from the outside, and then there's gonna be the microscopic, which is when it's like zoomed in, that's like the nephron. So the macroscopic view, you can kind of picture, I'll draw like a little mini one. So I don't take too much of my space here. But you can picture what we just went over, right? You have your cortex. The cortex is that outer portion. The medulla. The medulla is made of pyramids, right? Each of those little red guys would be a pyramid. And then together they flow into the renal pelvis. And the renal pelvis, first, what we get is we have what I was explaining to you guys before. First, it's the collecting duct. Then it's the minor calyx, we'll just put calyx. Then those flow together into the major calyx, then into the renal pelvis. Then once in the renal pelvis, right here, it flows down into the ureter. Our ureter is connected to our bladder, E for bladder. And then our bladder is connected to our urethra, which is either in the penis or basically just passes through to the outside for females. So here's my urethra. So the urethra has 2 different sphincters, which you do need to know. So I'll kind of draw them big so you can see them, the blue and the green. So blue would be your external urethral sphincter. This is made of skeletal muscle and it's voluntary. So this is like when you go sit on the toilet and you go pee, right? You would be using your external urethral sphincter. Versus your internal urethral sphincter is actually made of smooth muscle, and this would be involuntary or subconscious. So when you feel like you need to go pee, the smooth muscle internal urethral sphincter has relaxed, and now you would use your external urethral sphincter to actually voluntarily go pee. We know that the urethra is the actual structure that drains the bladder. The bladder has a little top portion, kind of like the stomach, and this is called the fundus, top portion of the bladder. A few more macroscopic things. The kidneys are outside the peritoneum, and we call that retroperitoneal. And then the right kidney is lower than the left. So it's just kind of stuff that's like easy test questions that you would want to know. You know, right kidney lower than left, kidneys are behind the peritoneum. Peritoneum is that covering around them, the abdominal cavity. Okay, so microscopic. Microscopic is dealing with the nephron, right? So the nephron and the function of the kidney is for filtration. So what is it filtering? It's obviously going to be filtering blood. And what is the location of filtration? The location of filtration is the glomerular capsule. I forgot that name for it. I said Bowman's capsule and renal capsule, but it's also called the glomerular capsule. So it has one more name I didn't include earlier. So renal corpuscle, Bowman's capsule, glomerular capsule. So 3 names. So filtration happens in the glomerulus. So that's one part of the nephron function. Then we have reabsorption. Reabsorption is taking back and reclaiming kind of what too much was filtered. So this is going to connect from the renal tubule to the blood. And the majority of reabsorption is occurring in the proximal convoluted tubule. Not all of it, but most of it. Secretion is from the blood back into the renal tubule. This is like if too much was reabsorbed, we would put some back, and this is the majority happening in the distal convoluted tubule. And we say CT, referencing collecting duct. Okay, so that's like your little, I guess, outline overview. And then obviously we were going to draw a picture. So like blood, essentially this is just saying blood is filtered out. Blood is filtered through, blood moves through. After blood gets filtered, we get urine. So that the end result here after filtration, reabsorption, and secretion is urine. But when we go through filtration, that's not urine yet. I already told you that there are 2 types of nephrons, right? We called them A and B in the little exercise we did. Cortico, juxtamedullary. Juxtamedullary is majority in the medulla. And then this is majority in the cortex. Okay, true or false? Both A and B have their loop of Henle in the medulla. True or false? So both cortico and juxtamedullary. Good, true, right? So that would be a true statement. So for the test, both have their loop of Henle in the medulla. It's just that the cortico majority is in the cortex, juxtamedullary majority is in the medulla. Very nice. All right, so that's the function. So this would be nephron. Oops, that didn't go well. Microscopic nephron. This would be function here. Now structure, the nephron has 2 parts. It has one, the Bowman's capsule. I don't know why the notes didn't start with this. And number 2, renal tubule. And then the renal tubule is in the proximal convoluted tubule, the loop, we'll put loop of H, and then distal convoluted tubule. So 2 parts to the actual nephron. Again, nephron would be the functional working unit of the kidney. For the test, that's its name, functional, or its definition, functional working unit of kidney. And again, nephron structure is in 2 parts, Bowman's capsule and renal tubule. So what we're going to go into now is drawing it and actually talking about how it works, because we can't really stay on the outline the whole time. But we're going to talk about filtration, reabsorption, and secretion next. So my goal is to cover filtration, reabsorption, and secretion, and then do the conditions and urine, like some components of urine. And then Thursday morning, we'll talk about the loop of Henle, which is called the countercurrent multiplier system, the renin-angiotensin-aldosterone system, and ADH, the 3 mechanisms that regulate blood pressure. So this lecture is arranged into the macroscopic view of the kidney, kind of topic number 1, microscopic, or the nephron, and we talk about that. Then we add on kind of 2, 3 more. There's conditions associated with the urinary system, and I'll go over those. There is the actual urine, like what is in urine, and we'll talk about that. And then there's 3 ways to regulate blood pressure. And we'll talk about that Thursday morning. Does that make sense? So urine and conditions, and then I'm going over microscopic right now. So if you're in the microscopic view, we're looking at the actual nephron. And we said that the nephron has 2 parts, right? First, it has that Bowman's capsule. Bowman's capsule, this would be part number 1. And then it has the actual renal tubule, right? Renal tubule. So that's number 2. So those are the 2 parts of the nephron. This here would be the proximal convoluted tubule. This is the distal convoluted tubule, PCT. Here is my loop. Penley. This would be my renal corpuscle, glomerular capsule, Bowman's capsule, which I already labeled. So what happens is you have your actual descending aorta, which is kind of what I labeled on the model. So here's my descending aorta. My descending aorta branches off into my renal artery. My renal artery then becomes my afferent arterial. Remember, afferent arterial actually flows into the kidney. And then flowing out of the kidney is my efferent arterial. So the efferent arterial leaves, afferent flows in. And remember, it makes this little capillary bed, but what do these capillaries have? They have these little holes, right, called fenestrae that are allowing stuff to leave. So this is fenestrae. These are fenestrated capillaries, and they're allowing stuff to leave the blood. So what is leaving the blood? So anything that can fit through the hole. So Knudsen talks about this, and I really think it's a good analogy. You know, if you make spaghetti, you boil spaghetti on the stove, and then you put the spaghetti in a strainer after you cook it. All the water flows out of the strainer, and the spaghetti stays inside the strainer. Does that make sense? And all of that water goes down the drain. So if you picture, I mean, honestly, it's not a horrible idea for us to draw it because it's literally the same thing that's happening inside the kidney. So here's all of my spaghetti noodles, right? And then it goes down the drain. Does that make sense? All that water goes down the drain, and the spaghetti noodles stay in here. It's literally the same exact thing. Blood cells stay inside. So what's left that actually leaves would be like red blood cells, white blood cells, little platelets. None of that stuff is getting out. But what's going through is like water and solutes will actually pass through and go down the drain, and down the drain is this way. Does that kind of make sense? So they're really a good analogy for each other. So things that would pass through that we like give you this example is you have like ammonia, which is something you don't want a lot of in the blood. So it's really good. We're getting rid of ammonia. And then you might have like glucose. You might have salt and potassium. Maybe you have like magnesium. So solutes, you could have salt.
Brooke?
Yeah.
I have a question. So when it's being unfiltered, is that the urine? Or no, this is not the urine part yet.
This is, it's like, I mean, honestly, I would say you could think of it like urine because it really is like urine, but technically it's only urine right here. So once it actually gets here, it's urine. All through the renal tubule, it's like your body is making it urine. Does that make sense?
So when the part that's like the spaghetti and it's being filtered, the liquid, or I guess the stuff that's coming out of that's being filtered, that's not the urine. That's just things that will still go through the entire system, and then they still have to become urine. They're not urine yet.
Yes. I think it would be probably better for you to think of them as urine, because really it will be urine in just a second. So I would tell you to think of it as urine. The only reason I'm saying don't think of it as urine is technically not all of it will leave the body. Some of it will be reabsorbed, just a little bit though. So like if you say like, let's say it's 100% represents all of it, right? 90% of it is urine, 10% of it will be reabsorbed.
Okay. So that means the urine is the solutes, or the solutes is what stays?
That all of this, so I'm writing them here. So this solutes and water. So the solutes would be like salt, sugar, ammonia, etc., and a lot of water. So
and this is what stays. So it does not become urine, or does it?
This will become urine. Yes, this will all become urine. Everything that fits out of the colander, everything that comes out of the colander will become urine. It's just that not all of it, but almost all of it will become urine.
And then the stuff that stays is what?
The stuff that stays, like you mean like this little part right here that I drew?
The stuff that's not drained.
Yes, the stuff that's not drained, like the noodles. Yeah. The noodles represent your red blood cells. You don't want to pee blood, right? Your white blood cells, you don't want to be peeing out your immune system. Platelets. So, yeah, not all of it filters out. And that's the whole thing about this. The next part of this is if you lost all water, you would die, right? If you lost all of your salt, all of your sugar, you would die. So what's weird about this system is all of it does leave, which is why I'm saying it's like it's urine, but it's not, because you lose all the water. Obviously, you can't just lose all the water and let it all become urine. You need to keep a little bit. So what your body does is after it all flows out, it grabs back what it needs, and that's called reabsorption.
Okay.
So like if you have your blood vessel here, reabsorption will take, I'll try to draw some little dots because I feel like the dots kind of help. Reabsorption will take some of those and bring them back into the blood, because they're like, okay, well that didn't go well. You know, we didn't want to lose all of the salt. Let's bring back some of the salt. We didn't want to lose all of the glucose. Let's bring some of it back. We didn't want to lose all of that water. Let's bring some of it back.
Right. So the urine or whatever's getting excreted has to go through that system, through the PCT and the leukopenulae and the, okay.
It can't become urine unless it goes through this. This process, the best way to say it in like the most like normal terms is called concentration. It's like making your urine concentrated. So like somebody who, there's a condition like a patient could get where they don't do this, and what they basically do is they just pee out everything. It's like peeing water and all of their solutes. So they just literally lose, their blood gets really thick, right? They're losing all their water, they're losing all of their solutes, and they're just peeing it all out, right? So we can actually do this when we give a patient who has heart failure like a diuretic, right? A diuretic is like, okay, well, we don't want you to keep any of it. Your heart is overworked. We need to get rid of all of that. So it's like it forces your kidneys not to do their job. But your kidneys, to keep most of us that are sitting here, we don't want to just lose everything, right? That sounds like a really dangerous thing for us who are healthy. So we keep all of those in reabsorption, is pretty much bringing back like a bunch of it. And then at the very end, if for some reason you're well hydrated and you have enough water, sorry, I wrote the wrong thing. If for some reason you were well hydrated, you have enough water, if you had enough glucose and you didn't need it, if you had enough salt, What it does is it does the opposite right at the end, and it puts back anything that's extra. So it says, okay, I'm going to just secrete that. I didn't need that much salt. I didn't mean to take back that much. I don't need all that. Let's just put back the salt we didn't need. Let's put back, for example, these are just examples. Let's put back the glucose we didn't need. Let's put back any water we didn't need. But like the priority for your body is to keep water. It's only going to be putting that water back if you have enough. That's why if you don't drink enough water, your urine is concentrated. If you drink a lot of water, your urine is not as concentrated because your kidney's priority is to reabsorb the water and only put back water in secretion if you don't need it.
Right.
That makes sense?
Okay, this is probably like off topic, but for someone that like holds their pee, is that like enlarging the Bowen's capsule?
Yes and no. It depends on how long you're holding your pee. You know, if you're someone who's like, okay, well, I'm not going to go to the bathroom for 2 hours. All that it's going to do is just fill your bladder, and your bladder, because remember, all of this stuff is going towards your bladder, right? So your bladder is just going to get more and more and more full until you need to go. Now, what can happen if a patient can't go pee? So let's say they have a tumor, which does happen, like a cancer tumor, and they have a cancer tumor like right here. So what happens is the cancer tumor maybe is in their ureter, it could be in their kidney itself, it could be in their bladder. For whatever reason they cannot pee, then yes, exactly what you said will happen. It'll start to back up, and all of that stuff is still trying to filter through, but it can't. So now what happens, that's like you're asking great questions. Now what happens is the stuff that's supposed to be filtering out is getting pressurized back. So all of those solutes aren't being filtered. Instead, they're going back. So what happens is the person's blood pressure goes up. All of those solutes in the water can't pass through. And now it's like they're fluid overloaded. So they get fluid in their lungs. They're going to get fluid in their tissues. They're going to be edematous because they can't filter. So yes, kidneys backing up is something we see in the hospital all the time. It's not always because of a tumor. It could be because of any number of things. But yes, that is a major issue for your body. What its goal is to get all this stuff out, right? But keeping the good stuff, getting rid of the bad stuff. You know, if you go out and you drink alcohol, right? The body wants to get rid of all of those toxins in the alcohol and let that all leave, but it wants to keep the water.
So this is also related to the fact that when you take your blood pressure and when the bottom number is a little bit lower, it's because you're dehydrated.
Yes, it can be that if you have lower blood pressure, you could be dehydrated, because if your blood pressure is not high enough, yes, you could be dehydrated. Yeah, that's a possibility. It's not like something we see super commonly, but yes, that could happen.
Okay, thank you.
Yeah, no, great. Awesome questions. So this part over here, oh, of course I erased it. This part over here that we were all just talking about, that's filtration. That's step number 1. We filter the blood. Step number 2, like I was just saying, is reabsorption. You bring that stuff back into the blood. This would be renal tubule to the blood. And then in secretion, the blood sends back the stuff that it doesn't really want at the end of the day. Usually this is like, it took its time, your blood sugar was high enough, we don't need all of this stuff in the blood, and the sugar in the blood, let's put it back. That's why diabetics, right? People that are diabetics and they cannot use sugar because they don't have insulin. People that are diabetics have a lot of glucose in their urine because their body can't use all of the glucose. So it's secretion, it's high of the glucose, right? So the secretion stage. So reabsorption is going to happen 65% of it in the PCT, 15 to 25% of it in the loop of Henle. This is just important for you to know for the test. So almost all of it in the PCT, 15 to 25% in the loop of Henle. Reabsorption. Secretion primarily is going to happen in the distal convoluted tubule, and the CT references the collecting duct. So distal convoluted tubule and collecting duct secretion. Okay? Obviously, filtration happens over here in the Bowman's capsule. that make sense to everyone? So step number 1, filtration. Step number 2, reabsorption. Step number 3, secretion. We're going to secrete back what we basically what we don't want at the end of the day. Anything that gets into the collecting duct, urine is collecting duct and onward. So once you're in the collecting duct, the body can't go back and say, well, actually, you know what? Scratch that. We actually needed that glucose. No, once it's in the collecting duct, it can't, there's no take backs, no go back. So the section on the screen, is it urine? Yes and no. It will be urine when it's in the collecting duct. When it's in the renal tubule, we're working on concentrating it to make it urine. First, we reabsorb stuff just to make sure we aren't getting rid of too much after filtration. Then secretion, we give it back if we found out we actually had enough of that substance. Right? If you drink alcohol, the reason you pee so much is because it's a diuretic. So alcohol makes basically your kidneys not function right. And so that's why it's so dehydrating to drink alcohol, because your kidneys don't do a good job at keeping enough water. So it disrupts the system. That's alcohol is a natural diuretic, natural. And then we could give a patient a diuretic that forces this whole system not to work, and basically forces everything out. So these people lose their water, they lose their salt. It can cause a lot of issues for the patient when we give them diuretics that make them lose everything. Okay, so what is the name of this product here? It's called Filtrate. Filtrate is the name for the water and the solutes that pass out. Again, it would be anything that filters through the hole. Do you want blood in your urine? No, it's not supposed to be blood cells or white blood cells or platelets or protein. You don't want protein. Protein would be a really large substance. There are a lot of large things that are not supposed to be found in somebody's urine. But water and solutes is wanted in the urine as filtrate. Okay, so those are the 3 steps to the microscopic nephron: filtration, reabsorption, and secretion. Now we're going to talk about conditions and urine. So what's in urine and conditions of the kidney. So let's do urine first. So urine. What are our components of urine? So we already talked about the bladder. The bladder has the external urethral sphincter, which is skeletal, and internal urethral sphincter, right? I don't think I need to do that again. But going over here, one more thing with the bladder, I'll write it right here. Maybe I can just point to the bladder. Lining the bladder, you have what are called the detrusor muscles, and those help to squeeze the bladder. Squeeze the bladder. You also have rugae. Rugae increase surface area in the bladder. They're little convolutions on the outside. Convolutions on the outside of the bladder. So those are just kind of 2 little terms associated with the bladder you would want to know. So rugae and detrusor muscles. Okay. So back over here, urine. Components of urine. Components. So urine obviously has what you all know that urine has. Urine has water. Drink more water, your urine is not concentrated. Drink less water, your urine is more concentrated. But it also has 4 kind of key things that we want you to know distinctive to urine. So it has what's called urea. Urea is made of ammonia and carbon dioxide. So urea is found in the urine. It's what gives urine that smell that smells like urine. Ammonia and carbon dioxide. Ammonia is really not something that we keep. We just get rid of it. Creatine holds phosphate to create a rapid form of ATP. So when we create ATP, we convert ADP, which is an adenosine, and 2 phosphates, adenosine diphosphate. I'm telling you this because it's kind of like you can't really understand what it's trying to say without it. And when you do physio in the fall, you'll know this. And then triphosphate. So what creatine does is it helps add the 3rd phosphate. So as you can see, ADP has 2 phosphates, ATP has 3 phosphates. You could think T3, and the creatine adds this last one on. Normally, you have to go through cellular respiration. Creatine bypasses cellular respiration. Normally, students, not normally, but I have had in the past, students ask me, okay, was this the same creatine like for gym, like working out? Yes, it's the same creatine for working out. And then they say, okay, well, you know, I had one guy ask me, like, what do you think of creatine? And my opinion, and this is backed by like a lot of people's experience, you have to be very careful because the kidneys filter creatine. When you take creatine, depending on how much of a dose you're taking and how often you're taking it, it's putting a lot of strain on your kidneys. So are you taking a small dose with a lot of water? Okay, maybe it could be helpful to you. You're really helping your kidneys. Are you not drinking a lot of water, going to the gym, getting dehydrated and taking creatine? Okay, well now it's probably hurting your kidneys because your kidneys are having to filter all of this creatine. Obviously, it's ending up in your urine, right? So your kidneys have to do all that work. I know that I think it was actually Newton who had some kidney damage because of using creatine when he was younger, because it can actually damage your kidneys from making them overwork. So just be careful if you use it. You know, you want to make sure you're using it wisely. Uric acid. So uric acid is another component of urine. This comes from the breakdown of nucleic acids. So uric acid from the breakdown of nucleic acids, which are basically the DNA component. And then lastly, we have inorganics as the 4th component of urine. This is things like calcium, magnesium, sodium, and chloride. And then we'll put etc. All inorganics that we don't need, if we have too much, are going to end up in the urine. And then you also can get kidney stones, right? Kidney stones from an imbalance, a crystallization, or a calcification of any substance. So if you get too much of one of these substances, you would have kidney stones. So too much of, you know, anything, minerals, increased amounts of minerals, not enough water. Just know on your test, right, this could be any substance. So it's not just one specific thing. All right, so that's urine. So those are the 4 kind of components we want you guys to know. Obviously, you have water in your urine. You would have, you know, maybe some glucose in your urine. So those are things that can be in your urine, but these are the 4 that we want you to know. All right, so for conditions associated with the kidney, and whenever we finish these, we will end the lecture and the thing, and then maybe we'll do a few practice questions. So anuria. Anuria is without normal function of the kidney. Polyuria is too much, right? Too much urination. So an example of this would be with a diuretic. So diuretic, I'll put /alcohol. Alcohol is a naturally occurring diuretic. Oliguria. This is not peeing. So not creating enough urine. Too little urination. Like you said, right? If a patient wasn't peeing, what would happen? It starts to back up. Incontinence, this is someone who is no longer able to hold their urine. Sometimes they don't know that they peed. Oftentimes these are like people that are mentally impaired, and they they wet the bed at the hospital, but they don't know. They don't know that they peed. Suppression. Suppression is when the kidneys / we'll put nephrons because that's more specific. The nephrons don't even make urine. So when we're not even making urine all the way up at the nephron level, that would be suppression. This could be related to nephritis. Nephritis is when the nephron is not functioning. Another symptom that the nephron is not functioning, which I already told you guys, right, would be hematuria. We know that if we see blood in the urine, now we have a dysfunctional size of the fenestrae, right? Those fenestrae are getting too big because they're allowing blood cells, which I already said is too big. So the fenestrae are too large, right? And they're allowing those blood cells to actually pass through, which is obviously, that's not our goal there.
I feel like you might be sick.
Retention. Retention of urine is more like what we were talking about. This is you create the urine.
What's wrong?
Those nephrons are busy creating it, but you cannot release it. So you can't release the urine out. You're just creating it and creating it. You can't get it to leave. And obviously this would back up. This is typically found in males from an enlarged prostate. So that would be like the most common cause of retention that's like a natural cause. Cystitis. Cystitis is the buildup of cysts in the urethra and the bladder, more often in females. A few more. Dysuria. This is painful urination. Enuresis. Enuresis is the same as nocturia. Noct meaning night. Enuresis or nocturia is bedwetting at night, right? So you're uncontrollably doing this at night. This is usually when the smooth muscle internal sphincter, this is like in babies and children, is poorly developed. And so it's just involuntary that it just relaxes in a deep sleep and they pee the bed. Okay. So nocturia or enuresis, relaxes in sleep. And we already did hematuria. Okay, so would you guys like to do practice questions, a few before, for the next basically 10, 15 minutes? Or would you like to just end here and then we can do practice questions Thursday? So why don't you put option one, practice questions, some now, or option two, end here and do practice questions on Thursday. Okay. All right. So I will let you guys go in that case. Thank you so much for coming, and I will see you guys Thursday morning, 9 to 11. Thank you guys.